Eukaryotic growth and development is one of the most fascinating areas you can study, particularly human growth and development. Because if you think about it, everything to make a baby, a human baby, is actually in the nucleus, with the exception of a few mitochondrial genes in the mitochondria. Everything to make this is in the DNA. These are the recipes to make that. So if you think about it, at one point when you were growing in the womb, you needed to make a new arm, and you needed to grow some feet and start growing a brain. Well, you're not doing that anymore, right? You're not growing new arms. You're not growing new feet. How did that happen? The DNA is all there. It was needed for a certain amount of time. We don't need it anymore. Well, this all happens because the body regulates which genes are expressed at what times. The genes involved in making an arm were expressed at a certain time, but they're no longer expressed now. So in this video, we're going to talk about how all the ways your genes are expressed. Let's get started. So why would we need to turn genes on and off? Well, first of all, you probably know that each of your cells has a special function, right? So each cell of a multicellular eukaryote, that's us, expresses only a small fraction of our genes, right? They've got them all in there. They've got the entire recipe book in that DNA, but we're really only going to make a few recipes. We're only going to express a small fraction because these cells are generally specialized. So if you think about development, right, in our bone marrow, we have these stem cells that develop that will then, because they're stem, right, they're the base, the root, they will then differentiate and develop into many different types of cells, right? So specialization, development, we need to turn genes on and off. If you think about development, as I mentioned at the very beginning, right, you needed to grow an arm at one point, you don't need to do it anymore. So different genes are needed at different times or points in the life cycle of an organism. Afterwards, some need to be turned off, in fact, permanently, right? Like that growing the arm gene. We don't need to grow a new arm randomly. That'd be kind of awkward. Now, we also need to turn genes on and off in real time because of homeostasis, responding to an organism's need. We need to keep balance. Prime example of this would be insulin, right? Insulin is a protein that takes sugar and stores that sugar as glycogen. So insulin, the protein, occasionally needs to be made. Well, so how do we do that? Well, the body, in its effort to maintain homeostasis, will turn certain genes on and off in response to a signal, high blood sugar, low blood sugar, that it receives from the environment to know whether or not we need to make that protein. So if genes ultimately make proteins, well, genes can be turned on and off if we need that protein to be made. So I'm going to give you an overview of the whole presentation, and then what we're going to do is I'm going to go through each of these individual points in greater detail. Now, I will tell you up front, this is an emerging field of science, a lot of hot topics in this particular video that you're watching today. And, you know, in 5, 10 years or 2 years, some of these things may have changed. Our understanding of them may have changed. But this is my understanding at this point where we are in understanding how our body controls things. All right, so let's think about the beginning, right? So we got a cell, we got a nucleus, we got some DNA in that nucleus. Well, the first thing that you may remember is that DNA is wound around these proteins, these histone proteins, and then it coils, supercoils, and then it's all coiled up nice there in that nucleus. So one way of controlling whether or not a gene on this DNA is expressed is through modification of this chromatin, right? So remember, unless your cells are undergoing mitosis, they're not chromosomes, it's chromatin, right? It's just hanging out in these sort of coils. So if the coil is too tight, right, it's wrapped up really tight, that chromatin can't be accessed. So we can acetylate the histones, to open up so that we can actually get to this particular DNA. So one thing we can do is modify the chromatin. Another thing we can do is when it comes to transcription. So think back, right? DNA going to our messenger RNA is called transcription. And before we can make the RNA, we have to have all these things in place. Some transcription factors to tell transcription to happen, some enhancers to enhance that transcription. All of that you'll sort of see right here. Then, once the messenger RNA has been made, well, we need to process that messenger RNA. You may remember things like adding a 5' cap, a poly A tail, and then also alternative splicing. Meaning, if this is our original piece of mRNA, we can splice together different sets of genes so that we're getting some different outcomes. All right, so once the messenger RNA has been made, we can then go to translation. This is highly regulated as well. So this is regulated with little factors that start translation and then ultimately give us our protein, okay? Then we have a way of stopping translation, right? So let's say this was insulin. We were making this was the RNA to make insulin there. Here's the insulin being made there. What if we don't need any more insulin, right? Well, one thing we can do is actually degrade that RNA so it doesn't keep going back to the ribosome to make more insulin, okay? Then, once we have made this polypeptide, that needs to be processed into the sort of the three-dimensional shape of the protein. Lots of points of control there as well. And then in the end, if we no longer need the protein, in this case I've been using insulin all along, so if insulin's been made, then we no longer need it, we can signal for the degradation of that protein. All right, so here's what I want you to take away from this slide. Every step in the journey from DNA to protein is controlled by the body. And if all of these steps are controlled by the body, that means there's got to be points of control all along the journey. So there are lots of different things we can do at any process in this journey from DNA to protein to turn on or turn off these genes or let them be expressed. Okay, so let's start with chromatin modification. So control method 1 is chromatin modification in DNA packing. So the degree of the packing of the DNA, how tightly that DNA is coiled up in that nucleus, regulates transcription. If it's really tightly packed, right, no transcription will occur. If you think of you wanting to access something, maybe you need to access a certain book. Well, if you've got all these books thrown in some big room together and they're packed really tightly, you might not be able to access that book or access those recipes. So if everything is really tightly packed, you can't even get to the recipe. So we have two different ways to describe this. Euchromatin is chromatin that's been sort of loosened. This will let transcription happen. RNA polymerase can come in, transcribe these genes because it's open. However, heterochromatin is really tightly packed. So it's hard for an enzyme like RNA polymerase to come and get down in here because that chromatin is really packed tightly together. So in this case, transcription has been turned off. One way then that we can loosen or tighten that DNA packing is through histone acetylation. So acetyl groups can be attached to these histone proteins and turn the gene on by loosening it up. So let me give you an example. All right, so here's some open chromatin, meaning this is that euchromatin ready to be transcribed. If you look down here, notice that these histone proteins have been acetylated. See the acetyl groups? So the acetyl groups have attached to these histone proteins. That opens up this chromatin so that RNA polymerase can actually come and transcribe the gene. So what do these acetyl groups do? Acetyl groups attach to the histones, loosen up the packed DNA, and then essentially turn the genes on by allowing transcription to occur. So whether or not your DNA is acetylated in some points will determine whether or not that gene will be expressed. Okay, so another way to modify the chromatin is through something we call DNA methylation. DNA methylation is essentially turning off genes because we're blocking transcription factors. So, and this is general, there are some studies that show maybe methylation can increase in some particular areas of the DNA, but in general, our understanding right now is that the methylation blocks transcription factors. So methylation, turns off transcription. Let me show you how that works. So if you look here at a piece of DNA, we see our nucleotide, our nitrogenous bases here in the center, cytosine, guanine, adenine. Notice that the methyl group, which you may remember is just a CH3, is attached to these cytosines in some locations. What that does is it blocks transcription factors from being able to bind and therefore have RNA polymerase come and do the transcription. So if you're looking at my model down here, here's all our DNA wrapped around these histone proteins. If you look really carefully, it's kind of hard to see. The methyl groups are attached to this DNA at the cytosines. This then doesn't allow RNA polymerase to come in and transcribe those genes. In fact, when you may remember females have 2 X chromosomes, or many females do. If one of those is inactivated into a bar body, right, only one's expressed. We talked about this in a different video. This is because of methylation that helps make those bar bodies. Okay, so control method 2. So we talked about control method 1, modify the chromatin, either with acetyl groups or methyl groups. The second method of regulation is transcription regulation. So we have some regions on the DNA. We have some enhancers and we have some promoters. Let's talk about what those do. An enhancer, what you see here, is a distant control sequence, meaning it's different from where the gene is. So if you notice, here's our particular gene that we're transcribing. But farther down the DNA is something called the enhancer region. This is where activator proteins bind to enhance the rate of transcription. You will also notice we have a promoter region. This is where RNA polymerase and transcription factors bind to help the rate of transcription take place. So we have enhancers really far downstream. We have promoters pretty close to the gene we're transcribing. Both of these are ways of control. So for example, if you didn't have the activator, that's going to be less transcription. If you don't have the transcription factors, no transcription. So this is a way of controlling whether or not transcription will occur where RNA polymerase makes this messenger RNA. So here's a model of how that works, this little enhancer loop. It creates what we call this loop right here. So remember, here's my DNA, that's the blue, all right, all this blue, that's my DNA. You see the histone proteins. If this is the gene I want to transcribe, okay, let's say it's the gene for insulin. Well, what happens is the enhancer region, which is pretty far down, is connected in a way by these transcription factors to the promoter region. So what happens is this sequence and this promoter sequence here have all these factors bound to them, creating this sort of loop situation, drawing attention to this whole conglomerate so that the promoter region will have RNA polymerase attached to it, thus transcribing the gene. Okay? So if you think about it, enhancer enhances transcription. All right, so control method 3. Once we have made the RNA, all right? So once DNA has coded for RNA through the process of transcription, so we've already done all those things, right? We've acetylated the histone proteins. We've made sure there's no methyl groups. We've gone to control method 2, and we've made sure all the transcription factors were there to transcribe. So now we've actually done the transcription. So here's RNA that's been made. Okay, so we got this long chain of RNA. Well, before we send this out to the ribosome, we may not want all these genes to be translated. So let's say we want gene 1, 3, and 5. Well, we can splice out 2 and 4. Therefore, we're left with 1, 3, and 5. That's what goes to the ribosome. That's what makes this particular protein 1. Okay? If instead we wanted 2, 3, and 4, we'd take out 1 and 5, get 2, 3, and 4 together here. So the idea is that we can alternatively splice this RNA, these different exons, remember exons are expressed, to create a family of possible proteins. Okay, now we're to 4. So we've opened up the DNA, we've transcribed the gene into RNA, we've spliced the RNA so that we get exactly the genes we want. Now we're ready to go to translation. Well, if you remember, translation is just the actual making of the protein from the messenger RNA. So think back to one of our last videos. We talked about the ribosome. You've got the large subunit, you've got the small subunit, and you may remember there's a start codon, AUG, which gets this whole thing started. So that would have occurred down here a bit to give you methionine. So this process is actually controlled by something called initiation factors. These are proteins that bind to the small subunit to initiate translation. Okay, so if this is the large subunit, here's the small. An example of one of those is eukaryotic initiation factor 1. So EIF1, there's the initiation factor, and you notice that this protein kind of binds right down here, and this helps initiate translation. Well, if there's no initiation factor, translation doesn't happen or doesn't happen as efficiently as it could, so we don't get the protein made. All right, so we need those initiation factors to help translation. All right, so now, once we have that messenger RNA made, so let's go back to my example. The lifespan of a messenger RNA determines how much protein is synthesized. In other words, if that messenger RNA is hanging around a long time, that means the ribosome is going to use it a lot, and that means you're going to make a lot of protein. So, let's take my insulin, for example. Let's say we've made the messenger RNA, which is going to code through the process of translation to make insulin. So we've made that, right? That messenger RNA goes to the ribosome, making a bunch of insulin. Yay, yay, yay. Well, what if we don't need to make any more insulin? We've got enough. Well, that messenger RNA is still floating around, right? It can go over to the ribosome and continue to make insulin. So one way of dealing with that is to get rid of the messenger RNA. Because if we degrade the messenger RNA, we're no longer able to make insulin. So one way that this happens is through something called microRNA, miRNA, which are single-stranded non-coding RNA that helps to silence RNA. So if I've got some miRNA here, it basically matches, it comes down to my target. Remember, that is the mRNA that's going to make insulin. That miRNA binds to the target mRNA, and now I have double-stranded RNA. That's horrible. RNA. Double-stranded RNA. That's not good. The body does not like double-stranded RNA, and it will signal for some things to happen, including messenger RNA degradation. Okay? All right, so we've made it through messenger RNA. Another example I want to talk to you about is siRNA. These are short interfering RNA. So same thing. If we want to get rid of that messenger RNA, they can label it, and it sort of is labeled with what's called a death tag for messenger RNA. These are short interfering RNA. Well, how does this process work? So remember, the first thing that happens is we've got this messenger RNA tagged for degradation. A double-stranded RNA is formed. There's our double-stranded RNA. Then a dicer protein comes along, binds, and cleaves it into small fragments. All right, so now here is this fragment, this siRNA, short interfering RNA. Now, what does that do? siRNA, then with the help of this protein complex, RISC, will bind to another specific piece of messenger RNA. Again, this is the messenger RNA to make insulin we don't want anymore. siRNA binds to that at the specific site because it has a complementary code, signals for degradation of that messenger RNA. Okay, this is a complicated slide, but what I want you to take from it is that there are ways of getting rid of messenger RNA so that we don't continue to translate that messenger RNA into proteins. Okay, so we've arrived now at control method 5. This actually is talking about what we do with the protein. We went through all of those steps to make a protein. And now there are many things you can do to the protein. It might need to be folded a certain way. We might need to add some sugar groups. We might want to target it to be transported out of the cell. So there's all these things that can now happen to the protein. So let me just show you a few little things, right? We can add some sugar groups. As you see here, glycosylation. That's hard to say. And then also we can target it, right? It might be lipidated. You might have all of these things that are on it for targeting. I want to draw your attention to this one right here, ubiquitination. So this is the process of actually getting rid of the protein itself. So remember, I've been using insulin all along as an example. So if we've made insulin, went through all those processes to make insulin, well, what if we no longer need that or we got too much of it in the bloodstream? We want to get rid of it. That's going to be through the process of ubiquitination, and that's what I'm going to talk about in the next slide. So in terms of all of these, I wouldn't worry so much about them. Know that they exist, but I want to focus in on ubiquitination. All right, so this is protein degradation. What is ubiquitination? Well, it is when ubiquitin, a protein, ends in IN, that is known as the death tag for proteins. This signals a specific organelle called a proteasome to degrade the protein. So let me show you what that looks like. So notice here, here's ubiquitin, here's a proteasome, remember these break down proteins, and here's some particular broken down protein. Notice that ubiquitin has been attached to this protein here. Well, what happens? Once the ubiquitin has been tagged to this protein, you will notice that, as you see here, this ubiquitin signals this proteasome to break down the proteins into what you see here. So a proteasome is like this protein degrading machine that when it notices ubiquitin, the death tag for proteins, it breaks down those proteins into all these little 7 to 9 amino acid fragments. Okay? So, I've taken you on a long journey, and I want to end with the timing and coordination of this, right? Because I've been saying, oh, it gets methylated, it's turned off, it gets acetylated, it unwinds, you get these transcription factors. Well, how is all of that stuff controlled? Well, all of this is very complicated, but a lot of this results because of sequential gene expression. And that is regulated by something called homeotic genes. Homeotic genes regulate the development by encoding transcription factors, okay? So we were saying earlier, these transcription factors have to bind so transcription can occur. Well, where do the transcription factors come from? Well, the homeotic genes encode for transcription factors. So I've pulled up one of our favorite organisms to study, the Drosophila melanogaster, the fruit fly. And it has several Hox genes that are expressed to know whether you make certain parts of the body. So during the development of a fly, right, at some point you need to make this part of the fly, right? At some point you need to make the abdomen. Sometimes you need to make the wings. All of those are actually controlled by Hox genes. These genes that are homeotic genes that regulate when these transcription factors will be encoded. All right, This has been a long journey. This is a tough topic. And to be honest, scientists are still figuring a lot of this out. So as you sort of wrap your head around this, I want to take you right back. Remember, what is this about? This whole video has been about the way in which genes get turned on and off. Not only the genes getting turned on and off, but once we make the messenger RNA and once we make the protein, how do we get rid of those so that these genes can be expressed or not expressed?